Good afternoon, everyone. This is Aisha Sunchodhary speaking. It is my pleasure to welcome you to the 7th of our 8-part series on innovations in oncology trial design. It is my absolute pleasure to introduce Natasha Rajicic, who up until recently was a senior principal statistician at Cytel. She's now the executive director of biometrics at Arrowhead Pharmaceuticals in Pasadena. And she's also teaches research design at Columbia University's Applied Analytics graduate program. Before coming to Cytel, Natasha worked for Pfizer, where she was a statistical lead for a late-stage endocrine products team. She also began her career as a statistician on the Phase 2-3 Clinical Trial Center for Biostats in AIDS Research at the Harvard School of Public Health. Today she's going to be speaking to us for about 45 minutes on study designs for oncology and rare diseases.
Thank you, Asha. Thank you very much. Hi, I just wanted to show my face at the beginning, but we'll turn the webcam off to avoid any technical issues. And as Asha said, I was at Cytel for the last almost 3 years as working in the strategic consulting group. And it is primarily those experiences that I will draw on during today's presentation. I just recently joined Arrowhead. It's only my 3rd week there, so not many experiences and examples drawn from there. I wanted to talk to you today about some of the issues that I've seen and came across during my consulting experience at Cytel, and actually at some of them coming from my Pfizer. I wouldn't specifically include them in slides, but in my commentary, you may hear some experiences coming from my Pfizer years when I worked in late-stage endocrine diseases, mostly rare disease in adults and pediatric populations. And as I was preparing this presentation, it occurred to me that while working at Cytel, a lot of examples that came to us and to me to help clients design their programs and mostly design their studies, they wouldn't necessarily come in and say, this is the rare disease that satisfies this particular definition in US or EU. It would come up in conversation as, well, this is rare or it's difficult to accrue, primarily problems coming from a small population to draw from. So the reason why I'm saying that is, while there are official definitions for what constitutes a rare disease in the US and EU regulatory, a lot of these issues and barriers and problems that are coming up may not be specifically for diseases that satisfy these definitions, but are kind of on borderline rare or just plain difficult to accrue. So sample size is a big issue, but there's other issues as well. Unknown medical history, natural history of the disease, or not established endpoints, not established drugs mechanism of action. So all of those issues I touch on during this presentation, not primarily only small sample sizes of the studies and how to deal with that. But of course, that's one of the main, as we all know, barriers for rare diseases. And in this presentation, the screenshot of the FDA guidance that I put here on the left, I think it's very nicely written. It's only 27 pages, which, you know, it's manageable to actually read it. And And I encourage you to look at it, even if you don't necessarily deal with rare diseases in your everyday practice, which I assume you do because you joined the webinar. But it kind of reminds you of overall clinical development best practices and issues in establishing effectiveness and safety for any drug, including rare diseases. So it's a very nice document. Okay, so following the definition of what a rare disease is and concentrating on the clinical development for treatment of rare diseases, reminder of what is so different or why do we even pay special attention to rare diseases, since, as I said, it's not like you don't have to establish effectiveness, evidence of effectiveness and safety like for any other drug in order to get approvals, to proceed through clinical development stages, to establish the drug on the market and with payers and so forth. But what is different and often lacking is the adequate description and understanding of disease natural history, pathophysiology of the disease, and the drug mechanism of action, which of course not specific to rare diseases. And on top of that, often unclear and something that needs further work is selection or development of outcome assessments and endpoints. And the reason why that is if the rare disease treatments or treatments for particular rare disease have not been studied in the past or scarcely studied, there's no established framework for what outcome assessments are accepted, what endpoints to look at. So that could be one reason. That's why I included that bullet point. Or even if the rare disease has been studied and there are established standard of care, there may be that your drug, as it's entering the clinical development, really needs a different endpoint. That's what's out there. And I will mention that a couple of times because I've seen that a lot where you are coming with a new mechanism of action or new paradigm in treating the rare disease or any disease really, and there is an established or there is a drug out there, other treatment therapy that is available, that maybe you have to include in as a control in your study, but the actual endpoint that you think will show the most differentiation and it makes more sense for measuring for your particular drug is not established yet. You have to establish it, defend it, and want to use it in your trial, rather than trying to fit a square into a square into a circle hole by using what's out there. It's already established, but it doesn't really match your intended effects that you're trying to see. So that's another difficulty that I see a lot in the clinical development of So I wanted to purposely start with something that's not how do we deal with small sample sizes, because that's, and especially clients coming to us and to Cytel and to statistical support, that's a lot of times that's the first and foremost concern. They would come to us and say, well, we can't accrue as much as we think it's maybe needed by simple sample size estimations or expectations. What can we do? And I wanted to purposely not start with that because that's kind of like the obvious problem. And I wanted to highlight some of the other issues that are also important. But of course, I will go over some of the designs that help with limited available sample size to draw from. Natural history studies are important for any clinical development because they define disease population, range of disease manifestations. They can help you define what are the subsets of the population that will have the most beneficial response to your drug, so establishing predictive enrichment. Again, going back to what I just mentioned about understanding what clinical endpoints, timing and duration you should use for your particular drug, and whether you should use any newer or validated existing biomarkers. So all of that can be one way, of course not the only way, is through natural history studies. I noticed in a lot of times natural history studies of some sort are already available out there. So it's not that you as a sponsor, as a company, even or you're not even planning to do them, but you can draw on the existing. And I don't know if it's the fact that I'm a biostatistician, so when people come to me, they're thinking, okay, what is the sample size? What is the sample size I need? Or what is the power I can have with this? And often I probe for what is the prior data and evidence that's available for the particular disease and drug. And one of the things that I ask is about these natural history studies, which clients are sometimes not prepared to talk about because they don't think that a statistician may necessarily need to know about them. But in order to tackle the issues in rare disease, you have to really bring in all existing evidence and knowledge to be utilized in the planning, in the design of the studies. And so do not, when you go to your statistician, when you talk to your strategic, to your quantitative strategist, give them all the information there is so that they can utilize it in the best way. So with that in mind, I actually wanted to give you first example. Which is, and of course, because this is coming from consulting examples, I will be very vague about the actual indication or treatment or anything that may reveal these clients that I had a privilege to work with. So I hope you forgive me about having some vagueness in these examples, but hopefully I can give you enough information so you get the picture of where I'm going. This first example is a phase 3 oncology study, and it's a single phase 3 in this rare, the solid tumor that is being studied here may not be as rare, but the actual mutation that is being targeted is very rare. So we have this first example kind of embodies both oncology and rare disease. So rare oncology solid tumor with underlying rare mutation that is being targeted by this specific drug. And what's also interesting is that this study is a single phase 3, single arm study, already accepted by the regulatory authorities across the world. And the study is planned as a single arm, meaning all people, subjects are only treated. There's no comparator, and the evaluation is to compare against the historical control rate of success in this population. So the primary endpoint is based on the objective response rate. natural history part here is subjects will be pre-screened for this particular mutation, and if they have it, they would be screened then for inclusion exclusion criteria for the main cohort of the study, for that one arm part of the study. If they could be previously treated, this could be their first um, um, treatment sequence as well. If they were previously treated, they would be that information will be collected, of course, at start. If they do not have the mutation, they are eligible to enter the control cohort, where they will not be treated with the study treatment, but they would be information would be collected either as in cross-sectional kind of at one point in time manner, and the information collected would be if they were previously treated, their first line response would be collected from the record, so retrospective cross-sectional study for those. And if they had not yet received at least one systemic anti-cancer therapy, then they have to consent to the collection of treatment outcome and additional follow-up throughout the study. So they're not treated with this experimental study treatment, but they will kind of remain in the study as in this observational part where their current therapy would be, the response to the current therapy will be collected at some point. And the reason for what the use of all that is that a subset of those controls will then be, with known responses to the first line therapy, will then be matched to the subjects in the main cohort in a blinded manner, of course, and with some established algorithm how to do that. And the reason for all that is to do these additional exploratory analysis that you see on the bottom right, is if to evaluate the effect of mutation on the first line therapy, treatment outcome of the first line therapy, demographics, characteristics of patients' mutation. So you will have the matched cases matched by demographics and other characteristics, and you will have responses to the first line therapy among those that were treated with the study treatment and among those that were not, that were treated with other therapies. And then by looking at those two groups, these exploratory endpoints can be addressed, such as also identification of prognostic factors for patients with a particular mutation, how would they respond to the first line therapy. So the primary endpoint of the study, of course, which is going to be registrational, is to evaluate objective response rate against the historic response. And I put in a parenthesis that this is done using the confidence interval as a primary method of analysis. Even you can say you're testing hypotheses of whether the null hypothesis is this historic rate, but the FDA will remind you that such inference is not accepted in a single arm study. You're really doing an estimate of the response and evaluating by observing the confidence interval. So as you can see here, that this natural history part of the control part was specifically designed for to augment the knowledge collected during the phase 3 study. And the regulatory agencies actually accepted this very positively because even though there were some phase one studies done in this population and for this treatment, there was still a lot of information lacking, lot of knowledge lacking in terms of this particular mutation. So they welcomed this kind of observational part of the study that to augment that knowledge that would of course then be submitted in the submission package and inform all kinds of activities, post-marketing, creating the label and so forth. Okay, so the obvious goal or the obvious issue in rare disease, what do we do with the reduction to reduce not the reduction sample size, maybe that's a little bit misguided title, but how do we deal with smaller of the populations that are available to draw study samples from? And some of the strategies would be focused on high-risk patients. Of course, you have to first understand who's a high-risk patient. And even that, if you're focusing on high-risk patients, that means that you're already subsetting your already small population, and then of course it may have consequences for the label, which you may not like. So that has to be weighted against pluses or minuses for that approach. One of the first thing as a statistician that I would ask for is there a continuous outcome variable that can be used instead of binary variable, because of course a lot of times that would bring you extra power in using the sample size you can deal with. Defining composite endpoints, so kind of trying to catch more events that way, is also another option, but of course not an option for every case and every disease. This is also true for the bullet number 4, longer repeated observation period, so more events can be observed, or changes can be captured at more event times, and can help in actually seeing the differences of any sort. And then use of adaptive randomization, or a use of adaptive designs in general, which I'll have a couple of examples of as well. You may know at Cytel, we a lot of times do come to us with help with adaptive designs in general. I didn't want to make too much emphasis on that during this presentation, but naturally a lot of examples that I can draw from are using some form of adaptive design. So adaptive randomization is one of the ways to deal with the limited sample size, because you maximize the number of subjects actually treated with the test treatment. So you, in that way, can draw more information from your sample size. One of the obvious ways is probably adaptive dose selection, and this schematic is taken from the reference is given in lower left level corner, but basically, instead of doing the traditional phase 1, phase 2, phase 3, phase 2, you do the dose ranging studies. You select the dose, and then the selected dose goes into phase 3. Kind of your typical learning and confirmatory phase of the clinical development. There are ways, of course, to try to combine the learning phase and confirmatory phase, not necessarily in separate studies, but in get them a little bit closer, and I'll give you an example. And with that comment, I also want to say that for the past 3 years, as I worked at Cytel and with a lot of oncology applications, a lot of rare disease, small sample sizes, clinical developments that are had all these barriers and issues with coming from small rare diseases or small sample sizes. What happened is I stopped thinking of a long time ago of clinical development as phase 1, phase 2, phase 3. I really started thinking of it as this is your establishment of safety phase, or searching for a maximal tolerated dose or something like that, along with finding the actual dose that could be defended and carried through. So kind of all that part of it is a part of the learning phase, and then confirmatory phase, either coming as a separate study or extension of the work that's already done. But I think what you understand what I'm trying to get at is phase 1, phase 2, phase 3 in my work for the past 3 or so years just disappeared. Yes, we still put on protocol synopsis. This is phase 1, this is phase 2 study. But really, when you think about it, when you look at the whole clinical development plan, I started using words like stage, or this is stage 1, and in this stage 1, we're going to learn this and this information that we're going to use in stage 2, or confirm stage 3. The whole traditional phase 1, 2, 3 just doesn't fit in my mind with these newer or more recent clinical development plans. So this second example is an example of adaptive dose finding, and this is from the actual study that I was not involved. This study was completed before I joined Cytel, but it was a successfully completed phase 2/3 study where the dose was not known, the right dose was not known at the beginning, and then during to design the study, several options were considered. One, of course, adjusting for multiplicity of testing or comparing each dose separately to placebo, which would require 80% power for this particular case, total of 520 patients. Then some of the other options that the team considered in the design stage were operationally seamless phase 2/3 study and inferentially seamless phase 2/3 study. And you see the actual sample size that each of these options would require. And obviously, as you increase from the classical just adjustment for multiplicity going down to inferentially seamless, the sample size reduces, the total sample size reduces, and the actual study did end up with inferentially seamless and with only 350 subjects in that study. It was successful and led to approval. But of course, some of those savings in sample size do come with complexities and additional things that are not as easy to implement, which is first of all the preparation for the design involves doing extensive simulation studies, design details being presented way ahead to the FDA, along with the operational details and conducting the interim analysis. To conducting the interim analysis and preventing bias, which is an important thing. And as I said, I did say that may be difficult, but at Cytel, folks have done that so many times that it may be difficult for an individual sponsor who have done it before. But I'm trying to say is there are people out there to help you and who have done this many times. Another thing to think about is with operationally seamless option, so the middle option there, you do get sponsor involvement permitted at the end of the stage 1 or trial 1 to make a dose selection. And if you're doing differentially, the seamless design phase 2-3, the dose selection at interim analysis may not, the sponsor's involvement may be limited or not permitted. And you have to weigh that against how much in sample size savings and duration of the whole program. Because if that's not some control that you can relinquish to the DMC members and to FDA sometimes, then that inferentially seamless phase 2-3 study is not the best option for that case. In the first, in the operational seamless, you analyze each part separately, whereas the inferentially seamless phase 2-3 combines the data from both stages and of course then has to implement the appropriate statistical test in order to account for the fact that you chose the dose in the middle and use the same data then to the end, so to reduce the type 1 error. But as you remember, you substantial savings in sample size, so going from 520 to 350 with increased complexity. But you have to decide which one is. There's no free lunch, so you can decide what's best for your program. I mentioned early on that one thing in rare diseases, development rare diseases, may be selection of endpoints. And what I meant there is I was thinking more of what is the particular endpoint is there a new assay that we want to develop in order to really measure what our drug is doing? Is there established treatment out there and established endpoints that we think we have to use but are not the best for measuring changes that we think we're going to see? Another option is related to selection of endpoints is switching midway between non-inferiority and superiority designs. So, a little bit different, but not too much different. So in that case, you would have the established endpoint, but you may not be as confident that you can establish superiority, but you may design the study to initially to show non-inferiority, but you don't want to avoid the chance to show superiority if superiority is there. So doing this interim analysis, where this particular interim analysis also incorporates sample size re-estimation. So increasing sample size to either show non-inferiority or superiority. But the main element that I want to emphasize here is chance of switching for availability for you to start with planning for non-inferiority, but if there's enough evidence to have an option to switch to superiority during the study, instead of kind of ending up with study and saying, well, too late now, we should have had superiority at the end. Another example that I wanted to include is now going back to oncology example, specific oncology example, is dealing with scarcity of prior data. And I've seen this a lot, and just recently finished helping a sponsor, a client, submit a pre-IND application and briefing document to the FDA, where they outlined their whole clinical development plan for a single phase 3 study that has there was no prior data in that population. So you can imagine how ambitious that was, but it actually was quite positive experience. And some of these, the example that I had in mind with this recent experience, actually, the design that I suggested they use for that phase 3 study with unknown prior information is coming from this particular example. So this was done by somebody else at Cytel, I think also before I joined. And the client came with a problem, late stage development of cancer immunotherapy. There was a problem with patient recruitment and scarcity of data. And data from recent small phase 2 trial was insufficient to reliably size the phase 3 study. And another standalone phase 2 study was not feasible. So the question was how to design a pivotal trial that makes productive use of existing data. And in that particular study, so 2 interim analyses were planned, but this first one is particularly interesting because it gives you a way to have an early look for futility purposes, and not a group sequential type of look, but using go, no go, Bayesian decision types rules to see if this drug should be, the whole development should stop early, and based on a surrogate endpoint, which in this disease was a progression-free survival. So the second interim analysis was for sample size re-estimation, and it was based on overall survival. So that's kind of a, I'm not going to talk too much about that. But the first futility analysis is what is another way for rare diseases to kind of have a early look, see if this is worth continuing. Be very, you can set the operational characteristics around the projected decision rule. You can say it will fail if it's really, really strong evidence that this is not going anywhere. So this example I used in this most recent example that experience that I had, where client came and said we want to do a phase 3 registrational study and we have no prior information about this, and I suggested we did have the sample size re-estimation part of it, but I also suggested this early look of futility analysis based on a go no go type of decision to assess futility. And the last, I do want to end soon because I want to leave. Yeah, so this is our last example. Important thing about population enrichment. And I have to admit, I did not design a study that has population enrichment in mind. That I just didn't have that experience at Cytel, but others at Cytel have a lot of experience about that. And so much that there's an existing webinar that you can access from, I think there was one yesterday, and then previously there are some older webinars specifically addressing population enrichment. The reason why it is attractive for rare diseases is trying to figure out what is the subset of population that has some unique, let's say, genetic profile or characteristic that, if targeted, have better prognosis of to better treatment outcomes that you can target. This type of design, of course, includes an interim analysis at which point you have a goal of selecting the subset of the population. This design is not limited to rare diseases. It is used in non-rare diseases. And this particular example, for example, from the New England Journal of Medicine is the disease that was presented here is some cardiovascular issue. So definitely not rare. But the but the approach is such that you, your goal is to try to see whether there's a subset of the population that would have better outcomes, and you want to use that information, that knowledge that you learn in interim analysis, to then subsequently have an option to reduce further accrual only to that subgroup that you think will have better outcomes. And that's what this design gives you as an option, instead of restricting accrual only to that subset from the start, and you don't need, and then you kind of you place all your bets on very specific subset of population. Or the other option is to accrue generally, or all comers, without the this focused look at the subsets, and then you may end up with a study that the effect is diluted, and you don't, and on average you don't see, you don't find statistical significance and solid evidence. So this is kind of in addressing those kind of issues. Okay, so in summary, the development programs for rare disease often lack adequate knowledge on disease natural history. I'll give you one example where, maybe extreme example, I don't think many will attempt to do natural history part or observation part as part of their phase 3 study. But just I wanted to show you how far you can go. There are many, in between solutions. Reminder: evidence of safety and effectiveness is still needed and expected, no matter what of the disease. But there is a flexibility in how the scientific judgment is applied when evaluating the evidence. As, for example, in that first example I gave you, we are talking about single study, single arm study that will be a registrational study. So that's definitely, it doesn't mean that the evidence is lower, but flexibility in how do you evaluate it. I mentioned just a few of the strategies that you can apply. There are many more available. Some of them, for example, Bayesian historic borrowing in both design and the actual conduct of the study, master protocols, basket studies, basket designs, and both of those, Bayesian historic borrowing and master program and basket studies, I do know that there's particular Cytel webinars that are available, recorded past webinars that already happened, and recordings are available on the Cytel website. And these are the references that were mentioned throughout the presentation. I'll just flash that in front of you. I will highlight the reference number 3, FDA rare disease guidance. When I was younger, and I guess I thought FDA guidances were such a dry material that do I really need to read all of it? And I don't know if they got better or I got better in reading them, but I really enjoyed reading this one. It kind of organizes your knowledge and points to some new things, and I highly recommend it. And with that, we come to an end, and I'll let Esha take over. Thank you very much. Oh, by the way, this is just a second. This is my cat, one of my 2 cats. The other one maybe you saw is sitting behind me, and this one is looking at East software, the survival endpoint module. I just wanted to mention that.
My goodness, he seems fascinated by East, as of course we all should be.
Yeah.
Yeah, thank you very much, Natasha, for that webinar. Um, Before we head to Q&A, I just want to make a few announcements. As I mentioned at the very beginning of this webinar, this is the 7th in an 8-part series on oncology innovations that Cytel is running. The 8th webinar will be 1 week from today at 11 a.m. Eastern Time, and it'll be on oncology and the use of estimans. So our strategic consulting unit is putting together a panel on estimans and oncology. We look forward to having you attend. Also next week, we're starting a new webinar series on an introduction to complex innovative design. So this will be a webinar series where we have statisticians Cyrus Mehta, Thomas Burnett, Chris Jennison, Nitin Patel. They're all going to be given an introductory master course on their specific design preferences. So if you have younger colleagues who want an introduction to complex innovative design, or if you want a refresher course, please sign up for that webinar series as well. It will start next week and go through the entirety of the summer. Finally, I want to remind you all that you can still participate in this webinar by asking questions in the panel at the very bottom. With that, Natasha, can I give you your first question? Are you ready?
Please. Yep.
Excellent. So the first question is, what do you do with patients as they continue to accrue in the operationally seamless design as you are making dose selection and other decisions? And relatedly, would the speed of accrual impact the choice of the design?
Let me just try to understand. So what do you do in operationally seamless design? Oh, I think I understand. So in operationally seamless design, that's the option where you do have an interim, let's say interim analysis to pick a dose, and you of course do it on a specific time point until predefined number of subjects have been accrued. But I think the question is, what do you do for if the accrual is continuing? What happens to those additional subjects? And because in the operational seamless, you are using each stage as a separate. So you're using that first stage to select the dose, and then the second stage for evaluating the dose. So you either have to be careful at you most likely want to stop accrual to kind of have the two separate stages. That is not the case for inferentially seamless, because in inferentially seamless you're using information from both stages, and you can really use the information from both. For operationally seamless, for the second stage, you do want subjects to be randomized to the selected dose. So if you continue accruing to all the doses, you're going to end up with subjects who will, of course, contribute to safety, but may not necessarily end up on doses that are actual chosen doses. And so it would be in a way, quote unquote, waste. It's never a waste. So one option is to temporarily stop accruement until you choose a dose. Or in some way slow down the accruement. I can't think of any other. I'm thinking here. Operational seamless. You do have two stages, and the reason why this whole interim analysis and concentration on reduction of bias if you're going to say that you're going to use first stage for selection of the dose, and if you're going to have enrollment in that, that is what you cannot then use that data for the second stage, if you had enrollment as a sponsor in the selection, because you otherwise the conduct of the second stage may be influenced by the knowledge you got from the first stage, and that is a one thing. The first and foremost thing that we want to avoid is introduction of the bias and changing that estimate that you started with your study with, because you something changed as a result of the interim, and then at the end you don't know if your conclusions are really coming from what you designed at the start, or for some intercurrent event that happened to the study. So yeah, the short answer is your safest bet is to stop or pause enrollment until you select the dose, and then continue enrollment only in the selected dose.
Thanks very much, Natasha. The second question is, how do I know if it would be beneficial to do a natural history study? Are there certain characteristics in a given context that I should be looking out for?
Oh, that's a great. I mean, all great questions, of course, but I haven't thought of that. That is why. Um, so drawing on that example that I gave you, the sponsor actually wanted to do separate natural history study, but then decided to do this combined because it was so hard to find subjects to begin with. So they wanted to maximize the utility of the patient population. But the question of whether to do a natural history study, if it becomes so you when you start your development program, and ideally you start with in my mind with that label in mind, like what are we trying to show here? What is the population we are targeting? What is the effect we are looking for? What is the differentiation against standard of care or placebo we are looking for? And working backwards, then you're looking at, okay, what is all the unknowns that I should fill in order to go from the start of development till that label that's accepted and utilized. And if those gaps in your knowledge include what is the population I should target, what is the how does this disease look like over time? Should I target early manifestation, late manifestation? Where is the maximum benefit I can show with my drug? If you're not sure what endpoint to actually concentrate on, that will show the most differentiation from alternative therapies, or to show the effectiveness of your drug. If all of those questions are unsure and unanswered, you may think of natural history study, which can be done in parallel with. It's not like you have to do it first and then go into phase 1 first. It can be done in parallel with your early, and that's what the guidance says as well, with your early development studies, and use that study as kind of like your this helper to answer some of these gaps in your knowledge that you don't have, so that you can better select that endpoint for phase 3, that you can better select which population to target. Yeah, that would be my short answer would be, if you have too many gaps in knowledge, to select the population, to select the stage of disease manifestation, to select something, if you think it can help you guide in selection of the dose. If you think you need to establish a new endpoint that hasn't been used before. And if there's too many of those questions unanswered, then I would seriously consider natural history study.
Thanks, Natasha. I think that was a really exciting answer. So they should be used in a number of different situations. It sounds like.
Yeah. Let me say, it's the word natural history sounds so. That's why I kind of pause every time I say it. It sounds like something in discovery and before and registries and all that. But but I've seen it. So I don't know, maybe there's a better name for it. Kind of like your helper, your information gathering study. I think that's what I'll use in the future. It's like the study that will help you inform your kind of main studies that are leading to registration. Yeah, just wanted to make that comment. I don't like the natural history sounds so epidemiology or whatever.
Okay, great. Thanks. No, that's important. If you have time, I have one more question for you. So last question. Given that sample sizes are limited in many of these cases, should we schedule interim analyses earlier or later if we want to consider switching to a superiority trial midway?
Yeah, so that's a very important question as well. And the timing of interim analysis, let's say for switching in non-inferiority superiority or for sample size re-estimation, it's all so, the way I look at it, so study dependent. From the experience, a lot of indications and diseases, if you look too early, the data is too messy. And when I say messy, it's too variable, and you can't really see clearly too early. But what I'm trying to say is it really depends on the population, on the study, on the endpoint, and you have to do simulations to evaluate the best timing. There's going to be, you give something, you lose something. Yes, you may have earlier decision time point, but you may lose precision. But maybe as an organization, you are willing to kind of make those bets. And it has to be informed evaluation of what do you gain by early look, what do you lose by early look, what is the variability of your estimates, what is the confidence you want to have in making early decision, how much weight you want to put on it. For some diseases, I've seen rare diseases where the treatment is very difficult and population is very hard to get to. And if the treatment doesn't work, we want to know early. We don't want to be dragging people into this difficult treatment, and yet it's unmet need and it's a dire situation and there is no other alternative treatment. So even that is better than nothing. But if it doesn't work, we want to, of course, cut it off. And then it becomes this evaluation. What is too early? What is and what is early enough to kind of cut the losses and establish utility. So short answer, extensive simulations and evaluation within the whole company or organization. All of your stakeholders have to be on board. Your regulatory, your discovery, your clinical development, to understand what are the pros and cons, what do you win or lose by different timing of the interim analysis.
Excellent. Thank you so much, Natasha. To our listeners, if you have any more questions, Natasha's Cytel email is on the slide. She's still one of our executive network consultants, so feel free to reach out to her or to any of our SMEs. Natasha, I want to say thank you so much for this really interesting webinar. I'm sure there are more to come. So thank you.
Thank you. Thank you all.